Good morning to our audience today. I would like to welcome you to Immunic's webcast to present the positive interim data from our phase 2 CALLIPER trial of Vidofludimus calcium in patients with progressive multiple sclerosis, reported after the market closed yesterday. My name is Jessica Brew, Head of Investor Relations and Communications at Immunic. I will also be the moderator on today's webcast. Speaking on the call are Dr. Daniel Fitt, our Chief Executive Officer and President, and Dr. Andreas Müller, our Chief Medical Officer. Please note, all participants will be in listen-only mode, and this event is being recorded. After today's presentation, there will be an opportunity to ask questions. If you joined the webcast via the Zoom platform, there are two ways to submit questions. You can either submit your questions in writing via the Q&A tool of the Zoom portal, or if you would like to speak with us directly, please use the raise hand function of the Zoom portal to queue your question. Before we begin, I would like to remind you that this presentation may contain forward-looking statements. Such statements can be identified by words such as may, will, expect, anticipate, estimate, or words with a similar meaning. And such statements involve a number of risks and uncertainties that could cause Immunic's actual results to differ materially from those discussed here. Please note that these forward-looking statements reflect Immunic's opinions only as of the date of this presentation, and it undertakes no obligation to revise or publicly release the result of any revision to these forward-looking statements in light of new information or future events. Please refer to Immunic's SEC filings for a more detailed description of the risk factors that may affect Immunic's results and these forward-looking statements. I would now like to turn the webcast over to our CEO and President, Dr. Daniel Fitt, to kick off the presentation of our exciting CALLIPER interim data. Daniel?
Yeah, thank you, Jessica. Also from my side, a very warm welcome. We published this extraordinary and outstanding data last night, which we believe can fundamentally change the way we treat progressive multiple sclerosis in the future. Before my colleague Andreas Müller presents the data, I would like to point out that there are currently only few treatment options for progressive MS. That would be mainly Ocrevus for primary progressive MS. For secondary progressive MS, there's virtually no real option. This makes our biomarker data particularly interesting for Immunic, but especially for patients with PMS and the treating physicians. With this, I would like to hand over to Andreas for giving some background information on PMS, the study, and to show the specific data. Andreas?
Thank you, Daniel. So let's start with a short review of the different ways of how disability progression occurs in the different subtypes of multiple sclerosis. On the left side of the slide, you see the two relapsing forms of MS, relapsing remitting MS and active secondary progressive MS, that are primarily driven by focal inflammatory disease and characterized by the presence of MRI lesions and relapses. It is, however, known from a recent meta-analysis of a large patient database that disability worsening in these two subtypes are driven almost equally by relapse-associated worsening, or RAW, and by progression independent of relapse activity, or PIRA, as you can see on the lower side of the slide. Immunic is currently executing a phase 3 program for potential registration of relapsing MS. The right side of the slide shows the two predominantly progressive forms of MS. Primary progressive MS, or PPMS, and non-active secondary progressive MS, or non-active SPMS, are characterized by an ongoing disability worsening without or almost without any MRI lesions or relapses being present. It is known from the same recent meta-analysis of a large patient database that disability worsening in these two subtypes are almost exclusively driven by progression independent of relapse activity, or PIRA. There are very few treatments available in these progressive subtypes of MS, as many merely anti-inflammatory drugs seem to no longer work or have much less activity in these primarily progressive subtypes. Only ocrelizumab is approved in PPMS, and no relevant treatment is specifically indicated for the FDA, by the FDA, for non-active SPMS. It needs to be noted that non-active disease in progressive MS only means that no longer focal inflammatory disease is present. No longer are there MRI lesion or relapses. However, the patient still accumulates substantial disability worsening. As many anti-inflammatory drugs no longer work or have limited activity, these progressive subtypes of MS represent the highest unmet medical need in MS. Medications to go beyond an anti-inflammatory effect are thought to be needed to effectively treat progressive MS subtypes. Next slide. Let's dive into a reminder about our fully enrolled phase 2 CALLIPER trial in progressive MS. In August this year, we completed enrollment of CALLIPER with 467 patients randomized 1-to-1 to 45 milligram of Vidofludimus calcium or placebo. The primary endpoint of this trial is brain volume change, meaning brain atrophy. The key secondary endpoint is confirmed disability worsening. Immunic has guided that the final top-line data of CALLIPER are expected to be available in April 2025, about 18 months from now. What we are presenting today are interim biomarker data for about half of the enrolled patients, a little bit more than 200 patients, up to 24 weeks of treatment. The interim analysis was done for the biomarkers neurofilament light chain, or NFL, and glial fibrillary acidic protein, or GFAP. Next slide. Yeah. This slide has the statistics of the total 467 enrolled patients in CALLIPER. On the left, you can see that the progressive MS subtypes included in this trial. About 60% of patients have non-active SPMS, considered the most difficult to treat patients in MS, and for which no relevant treatment is currently approved in the US. Patients with primary progressive MS, or PPMS, make up about 1/3 of the enrolled patients, and there are a few patients also with active SPMS as well. The interim analysis was performed by an unblinded statistician, and only our IDMC has seen the unblinded data. The study remains fully blinded and is continuing. Immunic received unblinded group-level data summaries, but no unblinded individual patient data. No clinical endpoints were evaluated in this interim analysis to protect the scientific integrity of the final analysis, and no formal inferential statistical analysis was pre-planned. The interim analysis also included a safety analysis, and the unblinded IDMC observed no new safety alerts and recommended to continue this trial without changes. So let's begin with the data regarding neurofilament light chain, or NFL, an established biomarker in neurodegenerative diseases. Neurofilament is a neuronal biomarker, which means the NFL peptide originates from breakdown of proteins that occur when nerve cells are injured or are destroyed. Neurofilament initially is released into the cerebrospinal fluid in the brain, but eventually can also be measured in blood. This biomarker is used in a range of neurodegenerative diseases. NFL is well characterized in multiple sclerosis and is found to be correlated with brain tissue loss. Interestingly, there are some newer data in progressive MS disease that make it a marker for more diffuse brain injury that dominates progressive MS in the absence of focal inflammatory disease. Now let's look at these data. This slide summarizes a recent analysis of neurofilament data in the phase 3 trial of ocrelizumab called Oratorio that led to its approval in primary progressive MS. The analysis only included patients that were on active treatment and followed them for 8 years on treatment. Since PPMS patients can have some level of MRI or relapse activity initially, the neurofilament values in these patients were re-baselined at 48 weeks. This ensured a progressive MS population free of focal inflammatory disease and any future disability related to focal inflammatory disease. After the re-baselining after 48 weeks, patients were then divided into patients with low NFL, labeled L here, and with high NFL, labeled high. labeled age. The red curve shows the proportion of patients with high neurofilament who experience disability worsening over the next 8 years of continuous treatment. And the green curve shows this for patients with low neurofilament values at week 48. What you can observe is that progressive MS patients with low NFL, in the absence of focal inflammatory disease, experience substantially less disability worsening events over the next 8 years. Lower NFL is correlated with lower risk for disability in PPMS patients. The take-home message is neurofilament is not only a marker for disease activity in relapsing MS, but it also predicts future disability risk in a progressive MS population without focal inflammatory disease. Or simply said, NFL levels matter in progressive MS. Let's look at the changes in NFL in the oratorio trial and recognize what the amplitude of effect the NFL from baseline was for the phase 3 trial of ocrelizumab that led to its approval in PPMS. The upper right graph shows the nominal neurofilament concentration, the absolute neurofilament concentrations in the placebo and the ocrelizumab group. In the lower right graph, these values are converted into percentage change from baseline. What you can see is that at week 24, the same time point for which we are reporting this biomarker today, there was a difference of 12.4% between the active and the placebo groups. Please keep that number in mind as an anchor, as available change from baseline for the only available therapy that is FDA approved in PPMS so far. Now I'd like to shift to our interim CALLIPER trial data that we're really excited to present to you today. In total, 203 patients were included in this analysis. These are all the patients who have both baseline as well as 24-week biomarker data available. On the left side, you can see that in the overall population, which includes all of the subtypes of progressive MS, we saw a 22.4% improvement of the Vidofludimus calcium group over placebo at week 24. We believe that this is a substantial and meaningful difference between active and placebo treatment in this progressive MS population. In fact, in a post-hoc statistical analysis, a statistically significant difference was found for neurofilament concentrations at week 24 between Vidofludimus calcium and placebo with a p-value of 0.01. If you then, on the right side, look at the subtypes of progressive MS, you can appreciate that this difference in neurofilament at week 24 was consistently shown throughout all subtypes of progressive MS. In particular, we saw a roughly 19% reduction for Vidofludimus calcium versus placebo in PPMS and a minus 20% reduction in non-active SPMS. As a reminder, the latter is a difficult to treat population with no relevant FDA-approved therapies available. In accordance with our previous phase 2 data in relapsing remitting MS, active SPMS patients who have presence of focal inflammatory disease or relapses show a good response in neurofilament on Vidofludimus calcium treatment of 43% reduction. However, the patient numbers are still very small in this subgroup. This slide sets the CALLIPER interim data into this perspective of historical study in the same progressive MS subtypes. In the left part, we display the data for primary progressive MS or PPMS. We already spoke about the oratorio approval study for ocrelizumab, which showed a spread of NFL values between the active and placebo at 24 weeks of 12.4%. In the CALLIPER trial, we have observed an 18.8% improvement of active over placebo at week 24. In the center of the slide, you will find the historical data for secondary progressive MS for both non-active and active SPMS. The phase 3 EXPAND trial of siponimod led to approval in active SPMS based on clinical outcomes. However, it was negative for the non-active SPMS group. In terms of neurofilament in this trial, we show that the 12 months value, this is the earliest time point, is available from the trial application, What can be deducted from these data is that the neurofilament effect in EXPAND was almost exclusively driven by the active SPMS population, where focal inflammatory disease such as MRI lesions and relapse are still present. When looking at non-active disease without such focal inflammatory disease, siponimod essentially has little effect on neurofilament. Mechanistically, it can be understood since the effect of S1P modulators is on active lymphocytes. In contrast, vidofludimus calcium with its combination of anti-inflammatory, antiviral, and neuroprotective mechanisms was able to show a substantial reduction in neurofilament in both the active and non-active populations. To our knowledge, this is the first time that such a substantial effect in neurofilament has been shown in non-active SPMS patients. Please also note that the patient number in the non-active SPMS population are meaningful, as most of the patients in the CALLIPER trial were in this PMS subtype. And I would like to point out again that non-active SPMS is the PMS subtype with the highest medical need and with no relevant FDA-approved therapies available. The right side of the slide shows comparison between our phase 2 emphasis data for vidofludimus calcium in relapsing remitting MS versus historical relapsing MS study to complete the picture. We are excited about the biomarker data and the consistent effect across on neurofilament in the overall PMS population throughout all subtypes of PMS, and also in other subgroup analyses which are available in our public filing. In particular, we see a substantial neurofilament effect not only in the non-active SPMS subtypes, but also in the non-active population throughout all subtypes. As I stated earlier, non-active in this context means that these patients continue to experience substantial disability worsening, but without showing MRI lesions activity or relapses. They basically have a chronic smoldering disease with no inflammatory disease present. This non-active population is known to be largely unresponsive to anti-inflammatory treatment. Vidofludimus calcium was recently confirmed to be a potent activator of Nurr1, a known neuroprotective target. We hypothesize that this potential neuroprotective mechanism may be the reason that, in combination with the drug's anti-inflammatory and antiviral effect due to DHODH inhibition, contributes to the substantial effect in the non-active progressive populations. So let's go to the next slide. Let's summarize again what we have observed for neurofilament in this interim analysis of the CALLIPER trial. In the overall progressive MS population at week 24, neurofilament showed an improvement for vidofludimus calcium over placebo by more than 22%. A similar amplitude of effect was consistently shown across all subtypes and subgroup analyses. The NFL effect, shown especially in the non-active SPMS subtype, reinforces the neuroprotective potential of vidofludimus calcium in the absence of focal inflammatory disease. The data from this CALLIPER interim analysis are comparable or numerically superior to the best historical data available in the same patient population. And a previous analysis from a large pivotal trial in progressive MS population has shown that lowering NFL numbers may be relevant to translate into clinical endpoints in the final analysis of CALLIPER in April 2025. So let's also move over to the other biomarker in this interim analysis, GFAP. For GFAP, much less data are available and has to be considered a more exploratory biomarker. It is not a neuronal biomarker, but thought to be an astrocyte marker and will therefore reflect diffuse glial activation in a chronically active brain disease. And given that GFAP is not related to focal inflammatory or neuronal disease, its blood concentrations are found to be much more stable, and the amplitude of changes during treatment are lower and take more time to develop. Nevertheless, in the scientific community, GFAP is believed to be potential interesting biomarker for smoldering disease that is suspected to play the major role for disability worsening in progressive MS. The next slide summarizes the GFAP data of this interim analysis. On the left side, you see the overall CALLIPER population from this interim analysis, showing a trend for an advantageous differentiation of active over placebo at 24 weeks. As you just heard, the rate and amplitude of GFAP changes are historically less. The right side of the slide displays the subpopulation of CALLIPER patients in this interim analysis, who at the time had also already had 48-week GFAP values available. There seems to be a trend that the advantageous difference of active over placebo treatment becomes greater over time. In general, the GFAP data at week 24 are more immature than the NFL data, however, seem to follow the same overall trend. In summary, the GFAP data of this interim analysis show an early but promising signal. It is known that GFAP changes during treatment generally evolve slower than for NFL and with lower amplitude. We believe that the data will further strengthen with longer follow-up times in the final CALLIPER data set. Now I will hand over to Daniel Fitt, our CEO, for some concluding remarks and outlook to future milestones. Thank you.
Yeah, thank you, Andreas. And as you can hear, we are excited about these unique biomarker data that give us and patients with progressive MS hope for a potential new therapy for this so far difficult to treat disease. Biomarkers show evidence that Vidofludimus calcium's activity extends beyond the previously observed anti-inflammatory effects, thereby further reinforcing its neuroprotective potential, paving the way for reading out the overall CALLIPER trial data on brain volume data of the full 467 patients, which is expected in April 25. What also excites us a lot is that Vidofludimus calcium is aiming to address high unmet medical need in non-active SPMS, where no relevant treatments are available in the US. Beyond this, the results of the interim analysis may inform the ability to potentially reduce PIRA events in the ongoing phase 3 ENSURE program in RMS. What are the next steps in the PMS study? In the meantime, 467 patients have been randomized and the study is fully recruited. We therefore expect the top-line results of the entire study in April 25. This also includes the evaluation of confirmed disability worsening and brain volume change. In parallel, we are progressing with our RMS phase 3 study. We are planning an interim futility analysis to be performed in second half of next year, and the first study, ENSURE-1, is expected to be completed towards end of 25. The data presented today continues to build on the foundation established by our phase 2 data on lesions, relapse prevention, and biomarkers in RMS patients, now adding an impressive signal of effect in patients with progressive MS. We look forward to continuing to build our data package as we look to future data readouts from our ongoing program in the next few years, including final data in the PMS population in April 25. With this, I'm handing the call back to Jessica for the Q&A session.
Yes, thank you very much, Daniel and Andreas, for walking us through the exciting CALLIPER interim results. We will now begin the question and answer session. As a reminder, if you joined the webcast via the Zoom platform, there are two ways to submit questions. You can either submit your questions in writing via the Q&A tool of the Zoom portal, or if you would like to speak with us directly, please use the raise hand function of the Zoom portal to queue your question. And our first guest today is Andreas Agerides from Wedbush. Good morning, Andreas. Please unmute yourself and go ahead.
Good morning. Thanks, Jessica. Congrats on these results. Just two quick ones from us. So how do you see these interim results increasing the chances of hitting the primary endpoint at the end of the study? And then given what we know about NFL and its correlation to relapses and MRI activity, how do you see today's results potentially de-risking the ongoing phase 3 studies? Thank you.
Andreas?
Yes, I think I should answer these questions. So I think, as we have shown with the oratorio data, there is a relationship for example, in this large PPMS trial, that the neurofilament data actually predict or correlate with future disability, which also I think makes the translation of the current interim data here in CALLIPER more plausible with the final analysis into other endpoints. There are data from other trials where we had seen that neurofilament and brain atrophy correlated very well. There's others where it didn't correlate. However, I think the general tendency is that it seems like neurofilament is really predictive of good clinical outcomes, which I think will at the end decide, I think here, whether this drug will be approved in the future. And second of all, I think, how does your second question was how these interim data de-risk the ongoing phase 3 trial in relapsing MS. So I think, as I mentioned early in my presentation, there was this last year the publication or meta-analysis was led by Fred Lublin that basically looked at more than 30,000 patients in clinical trials and assessed whether their disability worsening was connected to a relapse or whether it was independent of relapse activity. And the expectation was that basically in relapsing MS, the disability worsening is driven by relapses. To everyone's surprise last year, it basically came out that there was about 50-50 distribution between distribution being due to relapses and independent of relapses. And that the other outcome of this meta-analysis was that the usual and approved treatments in MS, they actually reduce very nicely the relapse-associated worsening, but they did very little to the disability independent of progressive activity, independent of relapse activity, progression independent of relapse activity.
Okay, thank you to both Andreas.
Yes.
Are you still there, Andreas? I think we lost.
No, I'm still here, but somebody came into my conference room here. Sorry for that. So I think what it does is, I think here in this progressive MS population, the disability is more driven by progression independent of relapse activity. And here, I think that is really, I think, the unmet medical need in relapsing MS. And the intent of this CALLIPER trial was right from the start that if you show an improvement in the progressive disease, that really means that we are also influencing PIRA in relapsing MS.
All right, thank you. And I'll hop back in the queue. Again, congrats on the results.
Yeah, thank you, Andreas.
Thank you. Our next guest is Tom Smith from Leerink Partners. Please unmute yourself and go ahead.
Hey guys, good morning. Thanks for taking the questions and congrats on the data. Two quick ones on our end. First, I was wondering if you could talk a little bit more about how your NFL data compares versus the Ocrevus experience. You obviously had what looks to be better placebo-adjusted reductions here, but I guess in the Ocrevus experience, it seems like the placebo patients there had a roughly 2.5% decline at 24 weeks versus your data set where placebo patients had a 16% increase. Were there, I guess, any notable differences in patient population or anything else that you can kind of call out there to maybe help explain some of the differences in interpreting the data set? And then secondly, I was just wondering, is there any opportunity for any additional interim analyses from this study that could allow us to track the progression of these biomarkers over time prior to the clinical advocacy readout becoming available in 2025?
No, thank you for the question. So I think for the second questions first, no, there are no other interim analyses planned. And also, I think we have a meaningful data set now with half of the enrolled patients basically up to week 24 here now, that this will probably inform the outcome of the trial and basically give us guidance for what the data should look like at the final analysis, which is only 18 months away. So there's no more interim analysis planned or even in the thinking. The first question was a little bit the difference in the placebo patients in the oratorio trial of the PPMS patients as compared to our placebo patients? So I think this is a very good question. And I think I thought about this as well, because I think that's very obvious when you look at the data. You have to understand that in the natural course of the disease, really usually over the long term, neurofilament will go up. And with increasing age of patients or humans, it will also go up. So I think it's usually in the short term, the change in neurofilament in a placebo group is often driven by how much activity, meaning, for example, MRI lesions, do these patients have going into the trial? And even if you try to enrich them for active patients, because that's done for some trials. In the oratorio trial, some of the criticism of the FDA during the review was that there were relatively, there's a relatively good amount of activity in these PPMS patients that were enrolled in the oratorio trial, meaning there were a good amount of patients who had MRI lesions and relapses in this trial. That was a little bit unexpected. So in a lot of ways, I think I would say the oratorio trial by itself was probably relatively active. So it's not that the majority were active because it's PPMS, but there's a good amount of active patients in this trial. And when you have active patients before the trial, for example, that tends to increase the neurofilament at baseline. But then during the trial, some of these patients will settle down. Just this is regression to the mean effect. So that if you have an active population, the NFL will go down maybe slightly in the placebo patients. If you start with a population that not is as active, you probably tend more to follow the natural course of the disease. So I think that, to my mind, is the explanation. Here, we, of course, we have to monitor our patients and all of the PPMS patients until the end of the trial, until 120 weeks of treatment, to really have the final numbers of activity. But so far, we haven't seen much activity of the PPMS patients in this trial. So I think this is the likely explanation. And then maybe for the baseline values that you have from the oratorio trial, please remember that this was a Roche trial that used the Roche diagnostic setup. It is known, and that's unfortunately kind of the problem a little bit with the neurofilament, that you have different measurement platforms. Almost everybody's using Quanterix, which also we have used here for this trial. But the Roche trial was done with the Roche instrumentarium. And this is known that if you would probably, if you measure the same sample on Quanterix and the Roche instrumentarium, that the Roche values are somewhat lower. And there are publications actually out there and some presentations on meetings. So in a lot of ways, that's why we also have reported this as change from baseline, because what every company does and what we have done as well, you measure all of the samples from the same patient in the same batch and run it at the same time, so that whatever the difference in absolute value is due to an instrumentarium, for example, between trials, you get the same change from baseline value. So the change from baseline values are much more reliable between trials than the absolute concentrations, because they're depending so much on the different ways and how we measure neurofilament, unfortunately, at this point.
Got it. That makes sense. All right. Yeah, I appreciate you guys taking the questions, and let me add my congrats again on the data.
Thank you, Tom.
Thanks, Tom. Our next guest here in the queue is Matt Kaplan from Ladenburg. Matt, please unmute yourself, and good morning.
Good morning. Thanks, Jessica. Just can you give us, in terms of a sense, what were the potential outcomes of this interim analysis and how these results, with respect to changes in NFL, compared to your expectations?
Yeah, maybe I take that question, because that is a question we asked ourselves a couple of times when we planned it. I think the overall goal in including biomarkers here is really to give ourselves, investors in the market, a chance to see where are things on track, and also to compare all the different subgroups. And when we've seen the paper this summer, I think this is a very good publication from about the oratorio study, we thought, okay, yeah, that's now a data point where we think we need to adjust ourselves with respect to that. And we are very happy that very much overperformed from that point of view. And therefore, I think and all the learnings Andreas has laid out in the presentation, we believe that NFL really has a predictive power, but also is the reason why we did the very detailed subgroup analysis, and even on those patients without any inflammatory activity, to really see, is the signal still there? And we are impressed that is really a consistent picture among all of these subgroups.
Okay, that's helpful. And can you talk a little bit about the safety profile so far you observed in the study, in this interim analysis in the PMS patient population?
So maybe that's a question to me. So we have not seen on a blinded level because I'm still blinded and everybody's blinded still for this trial, any new safety signals that was different from the relapsing remitting group. Or the other patient population that we have looked at with vidofludimus calcium. In addition, this interim analysis included an unblinded safety analysis by the IDMC, and they were able to see all of the individual patient data on an unblinded basis, and basically informed us back that we should continue the study without changes. So this usually, I think, means that they haven't found anything worrying in the safety analysis and just want us to continue the study as planned.
Okay, very good, and congrats on the results.
Thank you, Matt.
Thank you, Matt.
And the next one is Yungo Kwak from Piper Sandler. Please unmute yourself and go ahead.
Hi, this is Junggu speaking for Yaz at Piper. Congrats on the data, and we have a few questions. First is, if you could provide some color on slide 13, which showed only the overall PMS population statistically significant with the post hoc analysis versus subgroups.
You mean if that's the only analysis that we have done, or how the analysis was done? Specify a little bit what you would like to learn a little bit more.
Yeah, we were just wondering if only the overall population was statistically significant, or if you actually did some more analyses on the subgroups to see if they were also statistically significant.
Yeah, so to be quite honest, I think we had no statistical analysis pre-planned, and that's very common for interim analysis, because you can't power a interim analysis because you have really, I think, no way of powering an interim analysis. So in that respect, I think this was a post hoc, and given that usually you take the largest population initially for a statistical analysis, we really only have done one statistical analysis post hoc here, no other statisticals other than the overall population. I think we can do more. However, I think please be reminded that we have a little bit, we only get group level data here from the IDMC from us. We have no individual patient data, which makes some of these analyses a little bit more challenging because we're still remaining blinded and only very limited group level data were given to us.
All right, thank you. And then our second question was, could you remind us what types of data will be shared for ENSURE-1? And do you worry if p-values also could take a hit by sharing interim data as well?
You're talking about the interim analysis for ENSURE, or sorry, I missed that a little bit. What?
Yeah, for ENSURE for next year.
So the ENSURE interim analysis is very different from this analysis. There will be no group level clinical data or or biomarker data presented or being evaluated in the phase 3 interim analysis next year. This interim analysis for the phase 3, as we always said, is an interim analysis that basically focuses on sample size adjustment. So please remember, the phase 3 trials, the phase 3 program is based on a number of relapses that we need to reach for each study to have enough relapses to show a statistically significant difference between vidofludimus calcium and placebo, and on an assumption of conditional power or hazard ratio. And in a lot of ways, going into a trial, you make assumptions, for example, about how many relapses you collect from each patient in 72 weeks of follow-up. And by checking this, but also checking this through basically the hazard ratio and also the conditional powering of the study, you allow basically to adjust the sample size of patients to be enrolled to the number of events that you need to show statistical significance. So in a lot of ways, the study is driven by events and not by patients. And that's why we sometimes need to adjust the patient numbers so that we reach the number of events. So in that respect, it basically next year we would, on the phase 3 study, report only a sample size adjustment. And we also have a non-binding futility analysis done, which usually is part of this kind of sample size adjustment. So, but no group level data like here for CALLIPER.
All right, thank you very much. This is helpful, and congrats on the data again.
Yeah, thank you.
Thank you. And we have a couple of additional questions that came in writing, and I will just go through them one by one. The first set of questions comes from Madison Al-Sadi over at B. Riley Securities. First question, lower NFL correlated with lower risk of future disability in PPMS. Is this observation normalized by baseline NFL levels?
Yeah, I think yes. This was I think laid out in the paper we cited here in our deck.
Okay, the next question is, could you speak to differential NFL response across different subtypes and what may drive this mechanistically?
Yeah, I think that's what was the purpose of the analysis we have shown today. So there is a consistent reduction, as Andreas has shown, on the PR-related effect on NFL reduction, and on top of that, in the very small fraction of patients with inflammatory activity, you see a higher NFL reduction because there's additional effect coming from the anti-inflammatory effect of the drug. So I think it's the combination of both. It's, for me, it looks very consistent with the concept we are proving here.
Thank you. And then final question from Madison. Are you seeing NFL level change correlate with other efficacy endpoints?
So we cannot do this, and it is really a bit the setup of this interim analysis, yes, that we have to remain blinded because the study is ongoing, the study is not unblinded. We only get group level data, so on the individual, we don't have individual patient data, and we cannot correlate with anything on the clinical end here in this interim analysis. And this only has to do, we're not wanting to do, we would love to do it, but I think then you would unblind more or less the study. You can only do this after the unblinding of the study, after the final analysis. Here, I think with this interim analysis where we want to maintain blinding, it's impossible to do.
Thank you, Andreas. And then we have a couple of questions from an anonymous attendee, which I'm happy to read as well. Good morning, really nice update. 2 questions from me, especially as more literature is being published on these biomarkers. First, any comments on the baseline values of GFAP and NFL and what this tells you about the patient population that have been enrolled in both arms and the potential to see a meaningful treatment effect?
So I think I said a little bit about this, but maybe to reiterate it, I don't think we have too much data about GFAP and what the baseline means. So I think I have to defer this question. I can't answer that in some good sense. NFL, I think, of course, we have a lot of data, but as I pointed out, the absolute values that you often get as baseline values have to really looked at with the technology it was obtained, because they can be very different. And there is presentations and data available that these different technologies can lead to very different baseline values. I think the questions that I got before, that actually you're looking at the change from baseline for the placebo patient, is actually probably the best way to look at whether these populations were similar or dissimilar in the trial, because the more reduction you have in NFL from baseline in the placebo group, you would suspect that there was a lot of activity at baseline that led to that basically regression to the mean. And I think that's that's probably the best way of comparing these trials with these different technologies that we have for comparing or measuring neurofilament in blood.
Thank you. Next question is, based on what you have seen in CALLIPER, any thoughts on the patient population you will want to explore for phase 3? Could it be patients with non-active disease?
So I think the most exciting part of this interim analysis is probably that the non-active population, non-active SPMS, but also throughout the trial, the general non-active subpopulation of progressive MS patients really responded so well to vidofludimus calcium treatment. And I think we have presented is that this is where I think we see the biggest difference to previous trials. For example, I showed this Eponymous trial data, yes, that they did quite well in active MS and active SPMS patients, but did almost nothing with non-active patients. So I think here's the highest medical need. You have really no relevant FDA-approved therapies, for example, for non-active SPMS. So I think everybody would tend to say that if you work in non-active SPMS, that's probably the area where you have the highest medical need and where you should focus your further development. I think we have to decide it down the road with more data available, but I think based on this interim analysis, we're most excited about the non-active SPMS as the way to go forward once we have the final data.
One more question from an anonymous attendee, probably for Daniel. Could you talk about how this data changes partnership strategy and or alternative sources of financing?
No, it's always good to have good data. So therefore, it definitely is something which will help. And as Andreas just in the last question pointed out, seeing an effect in a patient population where so far nothing worked definitely may open different avenues to continue here development. And I'm very excited about that. And I see a lot of potential here going forward in these discussions, but the data is still warm. So we need to put that together and talk to the companies and talk to other players. And I think this really deserves a quick way towards approval. That's my personal thinking here, and maybe together with some other forces.
Yeah, thank you. And then I have a final question here in the queue. Is the company exploring preclinically whether the mode of action has applicability to other neurodegenerative diseases? If so, what indications appear most promising?
Yeah, sure. Of course, we are doing that. And this was, I think, we published earlier this year in Journal of Medicinal Chemistry that vidofludimus calcium is a potent, and maybe the only really potent Nurr1 activator so far known, with a relevant inhibition concentration range and amplitude of effect. And of course, if you dig into the literature, there's a lot published on the neuroprotective potential for Nurr1 activation and other indications. Some of those, for example, are Parkinson's disease and others, where there is already precedent from other studies, preclinical studies and mode of action studies. So of course, yes. And therefore, we also are looking on these other indications, but for now, we are definitely focused and prioritizing completion of this study and seeing the full clinical data as the first step.
Yeah, thank you. This actually were all the questions I had. If anybody has additional questions, please feel free to contact us via email or just call us. So this concludes our question and answer session. Thank you very much for joining Immunic's webcast today. The call has now concluded. You may now disconnect.
Yeah, and thank you, Jessica, for walking us through that exciting afternoon morning session event wherever you are in the world.
Goodbye, everybody.